my distinct pleasure to welcome Dr. Luis Herrea-Estrella, one of the pioneering a pioneer scientist in plant biology. I am sure you are all familiar with his work. As Dave mentioned, he's been the star of plant pathology lectures for the last 20 years. Am I right? Correct. For the following reasons. So Luis got his master's and both BS and master's in Mexico, followed by his PhD in Ghent, Belgium, during which, after which he also did a couple of years of postdoc in Belgium/Germany with Jeff Shell. During this period, he really pioneered work that it is the foundation of what we are doing now as plant biologists. He is the pioneer in using agrobacterium-mediated plant transformation and also selection of kanamycin as a selectable marker for plant transformation. And also during his time in Belgium, he identified a plant promoter, Robisco, and the regulatory elements that are sufficient and necessary in order to drive the transcription of general genes in plants. And also he identified the plant transit peptides, also using Robisco as a model system. And today he told me the story is a very interesting story. Given time, permitting time, he may want to share with you that story. If not, come tomorrow to Plant Biology Friday seminar, noon seminar. So today he's going to tell us about, where is my paper? You know what? Read it. That's what he's going to talk about today. And since his postdoc, he went back to Mexico to start plant molecular biology there. And now he is directing an institute for molecular genomics in Mexico. And given time, he's going to tell us more about it as well. Welcome. Thank you for accepting our invitation. And podium is yours.
Thank you. Well, I would like to thank Kathy and Bill Lucas for inviting me to come here to give a seminar. It was a surprise for me that Bill is not around. Probably, as always, he's traveling in Korea. Okay, good. So I'm going to talk about a topic that has taken quite a bit of my time in the past 15 years. I'm not an expert on the topic, but I've been dragged into many discussions about this thing. I want to start saying two things which are probably out of context during this seminar, but that I feel that are important. First of all is that I made small, but I consider significant contributions to the development of the technology to produce transgenic plants. I'm not ashamed of that. I'm actually very proud. And second, I'm not going to defend Monsanto. I have many reasons to have bad feelings against Monsanto. We filed the patent for agrobacterium transformation 2 weeks before Monsanto. After Monsanto spent over $30 million, they won the case. So we lost finally the tDNA patent. So I don't like the guys in Monsanto. So, having said that, I'm going to start my talk, and I would like to address 4 points. First, I'm going to give a very brief history of the development of transgenic plants. I want to apologize because I'm not going to mention everybody who made important contributions to the development because I don't really have time to go in all the details. Then a little bit about the commercial use of transgenic plants, public concerns on the use of GM crops, and the big problem of using transgenic maize in a center of genetic diversity. So what was the beginning? The beginning, it was the study of a biological model system, a bacteria that was able to produce tumors in plants that became a major problem for fruit growers in the late 19th century. It was in the beginning of the 1900s, in 1902, reported by the USDA as one of the most serious enemies of fruit growers. So that's where the study of agrobacterium started. And it was in 1907 that Smith and Townsend determined the causal agent of this crown gall formation. It was a bacteria, a gram-negative bacteria that was able to induce the tumors. They didn't know how they did it and so on, but they characterized the causal agent of this disease. Although there is a previous report from an Italian guy that unfortunately is written in Italian in 1897, where he first described white bacteria able to induce tumors in plants. But because it wasn't in Italian, it's not really credited. It was actually Armin Brown who did most of the early agrobacterium work. He did amazing contribution to understand the problem. He has several dozens of publications on this. And the first discovery that he made quite interesting is that the crown gall tumors that were induced by agrobacterium are able to grow in hormone-free media. So that means that they don't require the auxins and cytokinins to grow that normal plant cells need to reproduce in vitro. The second major achievement is that he could obtain axenic tumors where the bacteria was not present, and they kept their tumorous capacity. They kept growing in media without hormones, suggesting that the bacteria was needed to induce the tumor, but it was not required to maintain the tumorous state. Then he did very clever temperature shift experiments, which I don't really have time to describe. But using these shift experiments, he determined that the bacteria was needed only for a few hours to induce the tumor, and then it could be eliminated and the tumor was produced. He could use the bacteria for between 12 and 24 hours, kill the bacteria, and the tumor appeared many days after that. And in 1947, he proposed that this tumor-inducing principle was a chemical fraction of the bacterial cell capable of initiating, as in the case of the transforming substance of pneumococcus, DNA, a specific alteration in the host. So he was really predicting that the tumor was caused by the transfer of DNA, although he had no proof to do it. Interestingly, after 1950, he spent most of his time trying to demonstrate that tumor formation was not due to the transfer of DNA, but actually an epigenetic change in the plant cell caused by the bacteria. So that the bacteria produced some type of substance that induced an epigenetic change in the plant cells, and that was the reason of the tumor. Why he changed his idea is completely a puzzle for the other people working in agrobacterium biology at that time. But later, and he had to fight with this, there was a lot of support for his hypothesis of a genetic nature of the tumor-inducing principle. In Australia, Alan Kerr discovered that the virulence capacity could be transferred from a virulent strain to a virulent strain when they were inoculated on the same wound of a plant. If they were together, the virulence capacity could be transferred from one to the other. At that time, the conjugation phenomenon was recently described, so he proposed that probably the capacity to induce tumors was plasmid-borne because it could be transferred from one strain of the bacteria to another. Unfortunately, he didn't have the technology to analyze plasmid content at that time. And another interesting discovery was from Morel and Tempin, France, who discovered that the tumors produce substances that they call opines that were specific to the bacteria and not the plant species that was infected, not to the host, suggesting that the capacity to synthesize this compound was determined by the bacteria and not the plant. And since this was specific from the bacteria, they supported the idea that it was a transfer of DNA from the bacteria to the plant cell. Later on, my dear bosses, Jeff Shell and Mark Van Montague and Jean Nestor in Seattle, they made very important contributions. One is that all the virulent agrobacterium strains contain large plasmids that were absent in all the avirulent strains. So this was a correlative data, but it was very solid. They analyzed about 100 strains that were virulent, 100 strains that were not virulent, and they were all missing a large plasmid. Then using the Kerr's cross, this cross in which he inoculated the same wound with virulent and avirulent strains, they could demonstrate that when a virulent strain acquired the capacity to produce tumors, it also acquired the plasmid, making a formal demonstration that the plasmid was, in fact, responsible for the tumor induction capacity of the bacteria. Then there was a lot of work for the characterization of the TI plasmid. They denominated this plasmid TI because it's tumor-inducing plasmid, and they did by transposon insertion and a characterization of the different functional regions of the TI plasmid. So they discovered a couple of things that were quite interesting. One is that mutations in the plasmid alter the phenotype of the tumor, sometimes inducing root formation and other times inducing shoot formation, which led to the hypothesis that the bacteria was transferring to the plant genes that were responsible for the synthesis of auxins and cytokinins. And if you mutated the cytokinin genes, you get roots, and if you mutate the auxin genes, you get shoots because of the effect of cytokinins and auxins on development. So this suggested that indeed the bacteria was transferring a segment of DNA, but more importantly, that the transposons that were inserted in the tDNA were responsible for the phenotype, and therefore were co-transferred with the tDNA into the plant cell, suggesting that the tDNA could be used as a vector for introducing genes into plant cells. Very interesting. Then, of course, people started studying what were the requirements to have DNA transfer. And it was Maridel Chilton and Patrice Ambrinsky, who is now professor at UC Berkeley, who did the characterization of the junctions between the tDNA insertion and the plant chromosome. And they discovered that the DNA that was transferred from the plasmid to the plant was always flanked by 2 imperfect 25 base pair repeats. And all the transposon insertions in the tDNA were capable of producing tumors unless you remove the borders. So the only thing you needed to have transfer was the 2 25 base pair flanking regions, and whatever was in between is transferred to the plant cells. Then I worked with Patrice Ambrinsky and Kang Wan, who is now at Ohio State University, and showed that the right tDNA border is sufficient to direct DNA transfer and is vectorial. It depends on the orientation of the right border in which direction the plasmid is going to be transferred. Then the question came whether you could use the TI plasmid for plant genetic engineering. But there were a number of questions. First, whether the plant cells that contain the tDNA or other foreign sequences maintain totipotency. This question arose because Brown, when he was studying the tumors, could find that after some time of culture in vitro, the tumors started to form shoots. And he could graft the shoots into normal plants, but he never got transmission to the seeds. Whenever he got seeds, the seeds were normal and lacked the tDNA. So he suggested that the tDNA did something to the structure of what now we call the genome, and during meiosis, these cells were not capable of giving rise to germ cells. And then he proposed whether there was a mechanism by which the plant identified these foreign sequences and eliminated them from the DNA, from the chromosome during meiosis, and whether the presence or expression of foreign genes could disrupt gametogenesis or fertilization. The first, actually, probably the very first transgenic plants that were obtained were obtained by Patty Zambriski. She developed a TI plasmid in which she replaced all the genes involved in tumor formation by PBR322. Probably you don't know anymore, all the people younger than 40 years or 50 years maybe, don't know what PBR322 is any longer. PBR322 was the most common cloning plasmid at that time. So she replaced all the tDNA with PBR322, and she only maintained one of the opine synthesis genes. She transformed using an agrobacterium strain with this, what she called disarmed TI plasmid, on a wounded plant. Of course, there was no tumor formation, but if she took the cells from the wound site and put them to regenerate in vitro, she was capable of regenerated plants that produced the opines that were encoded by the TI plasmid, suggesting that the gene from the agrobacterium was transferred to the plant was still, the cells were still totipotent, she could obtain a plant, and the genes were expressed in the plant. Then through foreign genes, many attempts were done to detect the expression of the genes present in the bacterial transposons that were used to mutagenize the TI plasmid. They all failed, of course, because they don't have the sequences to mediate the transcription in the plant cells. So it was in 1983 that the group in Ghent, a group in England, and Monsanto, we almost simultaneously published the first transgenic cells that were expressing a true foreign gene. In all the cases was the kanamycin resistant gene. And this was the first time that Monsanto entered the scene. They never did any agrobacterium work or anything before 1983. In fact, they entered the agrobacterium world because Jeff Shell, our boss, suggested Monsanto that it was a technology that could eventually benefit a biotech, ag biotech company. In that time, they were a chemical company, not an ag biotech company. So in 1994, we produced transgenic plants and showed that the genes could be transferred to the progeny as any normal Mendelian factor, so that there was no problem of totipotency, germline transmission, or anything. And Monsanto, they also published their paper. As I said before, we published a few months before, and we filed the patent a few weeks before them, but we lost it at the end. Then binary vectors were developed in the laboratory of Rob Schilperud in Leiden, also a major contributor to the agrobacterium biology, which now is the plasmid you use to introduce DNA into plant cells. These binary vectors in which you have a small plasmid in which you introduce the gene, so your gene of interest, and you have a agrobacterium strain in which you have a TI plasmid that doesn't have tDNA any longer. So what is the conclusion? The technology was produced and developed in public institutions, mainly in Europe, Belgium, Holland, France, and Germany, with an important contribution also in the US by public institutions, the Washington University in Seattle and St. Louis, Missouri. Not Monsanto. Monsanto arrived at the very end. Then the applications started. And it was viral resistance in the lab of Roger Vichy, insect resistance in the plant genetic system, a Belgium company that was created by Marfan Montague, and some of us. Herbicide resistant, which as far as I know, it was Luca Comey who first developed these things and not Monsanto. He might be around here to clear myself. So they did it first, not Monsanto. I guess they got the patent rights or whatever they needed to. So at this point is when Monsanto starts to dominate the market. Because Monsanto starts to dominate the market, then we start to have problems. I think so. Ingo Potrico has been working with this project for the past 20-something years to produce rice with provitamin A. The lack of vitamin A is a major problem in many countries in Southeast Asia. Over 200 people is blind because of the lack of vitamin A. And although this is going to be delivered free of charge to the users, it's been impossible to transfer this to the farmers in the past 20 years. Now, there are even more promising traits like drought tolerance. Monsanto has important development for drought tolerance, as other labs in the world, but Monsanto seems to be closer to get commercial lines that could be applied. So how successful this has been? For some people, very successful. From 1996, in which the first commercial products were released into the market, to 2007, there were 120 million hectares grown worldwide. This is, as said by many people, is the most rapid technological incorporation into society. Much faster than electricity, much faster than telephone, much faster than cell phones. And the other interesting thing is that at the beginning, it was mainly developed countries who adopted this technology, mainly the US and Canada. Later, developing countries are almost reaching the same amount of land used for GM crops. And probably in the next 2 years, it will be higher in developing countries than in developed countries. Each time there are more countries that use GM crops. Mexico is here, but it's not really a major producer. But it's been growing and growing. And there are some very extraordinary examples. For instance, the Argentinian case. 100% of the soybean is GM soya. 70% of the corn is also transgenic. 50% of the cotton is transgenic. Is this good for a developing country? Absolutely, yes. Yield reached over 6 tons per hectare in soybean. The production cost reduced from $182 to $117. So they get more for less. In terms of herbicide cost, in 1994 they spent $78 per hectare in the herbicides. Now they spend $37 per hectare. And the use of insecticides has decreased 90%. So although the sales of glyphosate have dramatically increased, the total purchases of herbicides have decreased. How much the farmers have earned in this 12-year period? Over $20 billion. They are earning so much that the current Argentinian president, Cristina Fernández de Kirchner, created new taxes specific for GM soya and GM corn. Because they are making so much money, I said, you have to share with the rest of the people. Of course, the farmers didn't like it, and they opposed it. So I don't know exactly what happened at the end. I think that the president had to withdraw his proposal to create this tax. China is investing huge amount of money to develop their own products. They are afraid that Monsanto will sell them all their GM rice genotypes. India has been increasingly growing BT cotton from 28 hectares in 2003 to 6.2 million in 2007, and 3.2 million small farmers grew cotton in 2007. So this is not large mechanized farms, but the small farmers. The BT cotton decreased the use of insecticide in 70% and increased the productivity in 58%. So these are independent data. So the technology seems to work quite nicely, but this is really a global success? Not yet. Many countries don't like transgenic crops. Perhaps never will. Europe was going forward slowly, step by step, and now I learned that Germany is going back. No BT maize. Spain, that was growing already over 100,000 hectares, maybe next year is not growing anymore. So we don't know what's going to happen. There are still many public concerns. Many countries don't allow the cultivation of GM plants, including Mexico. Although as a tool for studying plant biology is a great global success. Almost every single lab doing plant biology uses the agrobacterium technology or the gene transfer technology to use their model systems. Then I have a question for you. Arabidopsis is a weed, am I right? It has all the characteristics of a weed. Short life cycle, produces an enormous amount of seeds. It grows very fast. Why we are not concerned that Arabidopsis might become a superweed? We are messing around with the genome of Arabidopsis in the most obscene ways we can imagine. And nobody's concerned. And I have seen outside the greenhouses of developing countries and developed countries, Arabidopsis growing outside. It's very difficult to control that these little seeds escape the greenhouse. And nobody is concerned. Why should we be concerned? Although it has all the traits to become a superweed. Not like tomato or corn. Have you ever seen growing next to the highways, invading the highways? No, but we are concerned what's going to happen with corn. Okay, so what are the potential negative impacts of using transgenic maize in Mexico? Of course, we have discussed this many times, human and animal health. I'm just going to mention a few things about this because I don't want to lose my time with this. Harms to the environment, but I want to mainly concentrate about this, the loss of genetic diversity of indigenous varieties and wild types due to problems caused by the insertion of a transgene in the genome of maize. Because this is now what academic people, scientists like you, argue that this is the main problem of using GM maize in a center of genetic diversity. So in terms of human and animal health, I just want to show you what the World Health Organization is called Organización Mundial de la Salud in Spanish. That's why it says OMS. And in fact, since Spanish is a official language in California, you should all be able to read in Spanish. So what they say, there is no evidence that the GM food could cause any harm to human or animal health. With the available evidence, with the available information to date, it can be concluded that the potential risk of GM food to human health are similar to those of conventional products. So this is after many meetings and after spending a couple of $1 million getting people together to discuss this matter. Of course, I'm not saying that every product is safe, but given the regulatory status in which each product has to be analyzed case by case, step by step, I think the system only will allow products that are safe to go through the process. So in terms of health, this is cotton growers in China. How much they spend in pesticides? The adopters, 31 yuan, the non-adopters, 243. How much they use? 2 versus 21 kilograms per hectare. How many times they sprayed? 0.73 on average. The adopters, 9 times the non-adopters. Health problems for the farmers. The adopters, the full adopters, not a single case of insecticide poison is reported. The non-adopters, 8.3% of the farmers went to the clinic because they had insecticide poisoning. So GM crops could have an important benefit for human health. In terms of ecology, Bt can have an effect on non-target insects. We all have to accept that Bt is not species specific. It is rather specific, but it covers a number of species, of insect species that it can kill. The question is this better? If we spray with a plain the insecticide and a totally non-specific insecticide? Not only for the ecology, but for this guy. At least 80 people dies every year in Mexico because of insecticide poisoning, because the planes just go and spray over the workers. They don't really care. They don't have time to lose because the plane costs a lot of money. Well, this I already said. Although the sales of glyphosate have increased enormously in the past decade, total sales of herbicides have reduced. So when people said it's because Monsanto is inducing an agriculture that uses more herbicides, it's true. It's partially true. They are inducing to use more glyphosate. But in total, the use of herbicides is being reduced. Now, maize production in Mexico. Between 2 and 2.5 million farmers grow maize as their major crop. 90% are small subsistence farmers. 60% are native Indians. And about 15 million people depends on the income of maize farmers. So this is an important issue. The 10%, this doesn't work anymore. The 10% of large farmers produce 70% of the corn in Mexico. So these 90% of small farmers only produce 30%. So the vast majority of corn is produced by the large mechanized farmers in the northwest of Mexico City. So we have an average of, thank you. We produce an average of 2.4 tons per hectare in Mexico. If we could increase from 2.4 to 3 tons, we produce about between 12 and 14 million tons of corn, but we consume about 20 million tons. So we have to import from the US 6 to 8 million tons per year. If we could increase from 2.4 to 3.2, we would not need to buy any more for corn from the US. The other problem is that we have to buy corn from the US, and it's GM corn. So we have been eating GM corn for the past 10 years. Nobody has died from eating GM corn, but we eat it. We cannot grow it, but we can eat it. So why is it problematic in Mexico? Because Mexico is the center of domestication and genetic diversity of maize. Maize was domesticated in Mexico about 10,000 years ago. For some still unknown reasons, there is a huge genetic diversity in maize, much more than other species. And most of the genetic diversity is in Mexico. And most of this genetic diversity is still used by traditional farmers. We have hundreds of different dishes made out of specific corn land races. If you want to eat pozole or tamales or uchepos or pinole or whatever, each one has a specific type of corn. So the question is GM maize a threat to maize biodiversity in Mexico? I don't have the answer, but what are the main factors that could reduce or are reducing maize genetic diversity in Mexico? The increase of commercial varieties. Nothing to do with GM varieties because they are not allowed in Mexico, just commercial varieties and hybrids. Migration to cities and the US and Canada. Globalization and free trade agreements. The question is whether transgenic maize plays also a role here. That's how much we have adopted hybrids. So every single state in Mexico is growing hybrids. So there is not a single hybrid-free zone in Mexico. So if the land races are living next to the hybrids, they must have already exchanged quite a few genes. And in some areas, 80% of the maize is hybrid. The least we grow is between 25 and 39%. All the other states have larger percentages of use of hybrids. So we are losing diversity because people is buying hybrids, and they abandon their traditional varieties because the hybrids are more productive, as simple as that. Migration. About over 300,000 Mexicans cross the US border every year. From where do they come from? Everywhere in Mexico, particularly Oaxaca, Chiapas, Guanajuato, from where I come from. The US put this iron fence all along the border. Didn't help. These guys are jumping the fence in the night, or they dig a hole and go through it. Or there is a sea of possibilities. You don't have to climb the fence. You just take a boat and go to the other side. In many rural areas, all the young men have gone to the States. So there is only children and elderly people left. So nobody to grow maize. So in most of Guanajuato state, there is no traditional maize being grown because all the guys from rural areas have gone to the States. So many that our local airport has now 3 direct flights to Houston, 2 to Dallas, 2 to Chicago, 1 to Atlanta, and 4 to Los Angeles. Not because we travel a lot to the States, because the immigrants are coming back and forth. So these are real threats to maintain maize biodiversity. Another problem, we have neighboring countries that grow GM corn. How are we going to stop gene flow? The iron fence is going to stop the pollen? I don't think so. Honduras is already fully going to GM. Next year, Guatemala and El Salvador. There is only a 10-meter broad river in between these countries and Mexico. How are we going to stop the gene flow? So the question is, can we contain GM maize from legally or illegally entering Mexico? How much time do I have? I think I have to hurry. 10 minutes? Okay. So gene flow between commercial varieties and land races has occurred for many decades. This is a fact. It's known that the farmers experiment. They go to their neighbors, they see something growing nicely, they bring back a few cups, and they plant, and they see what happens. They are experimenting continuously. The farmers actively select for the seed with the side characteristics. So they don't select by plant size or disease resistance or anything else. They mainly select for the type of grain. If you have a more healthy plant or whatever, they prefer it. But the main thing is they select for the type of grain. So, for instance, why the land races have been maintaining their identity? Because if she wants to make this, she needs this corn. She will not grow this, the American corn, yellow corn, because then you will get yellow uchepos, and we don't like yellow uchepos. Or if you want to eat pozole, many of you know pozole. We don't want yellow grain in the pozole. So they continually select for this. And it's not so difficult to do it, because if these are some of the land races. If you cross it to a hybrid, that's what you get. These guys are not stupid. If they keep some of these white seeds for the next cycle, and they grow better of what they had before, they will select for it. So for instance, if they incorporate the Bt gene, and once they recover the phenotype of the grain and they are insect resistant, they will keep it. If it's of no use, they will not keep it. So there was a few papers talking about the escape of transgenes in Mexico. And I don't want to say who funded my friend Ignacio Chapela, but he published a paper in Nature in 2001 saying that there was transgenes in land races in Mexico. In fact, he said that they were introgressed already. You know that introgress is when you have specific segments of DNA that have been fixed into the genome of a plant. That was a very statement, but he did. In 2003 and 2004, there is another paper from plant ecologists that don't like GM crops and did a serious analysis of the presence of transgenes in Mexico, and they said, to our bad luck, we didn't detect anything. Done in 3D independent labs. Now there is a more recent paper in which there is again detection of transgenes in land races in Mexico at a very low frequency, about less than 1% in 3 out of 30 locations. So maybe true, maybe not true, I don't know. I'm not going to judge these papers. But what we know now, the Secretary of Agriculture has declared that transgenic maize is being grown in Chihuahua, Mexico, intentionally by the farmers, illegally, and they had to purchase about 2,500 tons of corn that were transgenic. So we already have it there in large amounts. So these previous studies saying that whether it was 0.1% in, I don't know. Now in Chihuahua we have a huge amount of it. So the government was forced to allow experimental field trials with transgenic corn. After discussing this for 20 years, after having a biosafety law that regulates the release of transgenic plants in Mexico, which has not operated in the past 6 years, they finally said, okay, it's here, we cannot do anything. Make experimental field trials with transgenic corn in Chihuahua, because there is already transgenic maize there. So the gene flow is unavoidable. We have neighbors that grow transgenic corn. The immigrants bring back transgenic seed. So are there unexpected effects of the presence of transgenes in the land races? Is there something that we have to worry about? I make questions. I don't like to make. Could transgene introgression have a greater effect on the genetic diversity of land races or wild relatives than genes from hybrids? Do we expect that a transgene could have an effect that is completely different of what we could have from the genes of hybrids? Could the presence of a transgene alter the chemical composition of maize in such a way that it loses its identity? So that we introduce the BT gene in pozole maize and we don't taste pozole anymore? Will transgenes cause genomic instability in a different fashion than the naturally created than that naturally created by transposons or other mobile elements? Could we expect to have more unexpected pleiotropic effects from a transgene insertion than those caused by transposons that naturally activate or modulate the expression of neighboring genes? So many people have come with this idea of substantial equivalence. You have to demonstrate that your transgenic corn is substantially equivalent to a normal corn. And I said, okay, which corn? We measure the genome size of Mexican land races. And first of all, to which maize compare? Same size, different size. And to our surprise, there's a huge variation in genome size. Over 40% in genome size. That means that this one has 1.2 gigabases more of DNA than this one. There are some very interesting correlations. The lowland popcorn maize has smaller genomes than the highland maize that require more energy to grow at low temperatures. Very interesting correlation, but it doesn't matter here. Which one you use as a reference? Do they have the same number of genes? Do they have the same number of transposons? In terms of transposons, certainly not, because we did sequencing of this genotype, and when we compare with B73, there's a huge difference in transposons, including a lot of helitron transposons that I'm going to come back in a second. To what we compare? To a genotype grown on which conditions? We have done, I'm interested in studying what happens with a plant when you expose it to low phosphate, how the plant responds and how it adapts to low phosphate. So we did some experiments with maize. So we transfer the same genotype from optimal conditions to low phosphate. And we find that about 1,100 genes are differentially expressed, go up or down. So it's a huge difference between a maize grown on what we call optimal conditions versus low phosphate. What's the chemical composition of this plant? All the blue is that they have lower concentrations. All the red is that they have higher concentrations. Over 40, we measure about 400 compounds in these plants, and over 40% change their concentration, more than 1.5 times. 150%, 2.5 times. So should I compare maize grown under which conditions? Now, what happens between different maize grown on the same, exactly the same conditions, what we call optimal conditions, full irrigation, full fertilization, nice soil, and so on? We tested 3 different genotypes, doing nothing to them. 429 genes have differentially expressed over twofold between these 2 genotypes. 409 between these 2, and 252 between these 2, which are closely related. They are both derived from the Tuxpeño landrace, and although they are closely related, they express over 200 genes that are different. And what if we change the environmental conditions that we are growing these 3 genotypes? It gets much worse. We expose them to drought, because these 2 varieties are more drought tolerant than this one. This one has 611 genes that are turned on that are not responsive in these 2 genotypes. They share 246 genes, which is the common response of maize to drought, but there are a lot of differences. What's the chemical composition of these 3 genotypes? What if I compare them to land races that are really far? To what I should compare my transgenic? It doesn't really make any sense. So now, do the transgenes have an impact on genomic stability of function, this different to transposons? Or other factors? For instance, mutations induced by physical or chemical factors, aberrations in the recombination process, insertions of genes from organelle genomes into the nuclear genome, movement and amplification of transposons. In terms of transposable elements, the copy number can vary enormously from one maize genotype to another. This is a southern blot for AC. This I borrowed from Nina Fedorov. This is the line she used to clone AC because it had only one AC, and it was easier to clone. And she probed with the AC other genotypes, and you can have between 5 and 3,000 AC copies. So there is a huge differences. So can we expect effects different from what transposons cause? The transposons cause mutations by insertion and excision from genes. They activate and suppress transcription of neighboring genes. There are many examples how the insertion of a gene can shut down the expression or activate the expression of the gene. They alter transcription processing by introducing donor and acceptor splicing sites. They can move exons and promoter into existing sequences to create new genes. This is part of evolution. Transposition and amplification events of large pieces of genome that enlarge the genome size. This has been more carefully studied in the past few years. For instance, Hugo Donner sequenced a bag in which they had the bronze locus to see how different it was from between 2 genotypes. His surprise is that not only the number and type of transposons in the 2 exact same regions of 2 genotypes were different, but that the bronze locus of B73 was missing genes that were present in the other genotype. So they say there is lack of collinearity in maize, not like we expected before. So what they conclude is that we have found that the bronze genomic region of 2 North American maize lines differ extensively in the organization and content not only with intergenic retrotransposon clusters, but also of the genes themselves. And then they tested other genotypes. Is this an exception? Maybe B73, since it was used by the American companies, is a monster. So they did a pulse field gel for different genotypes, and they analyzed the same region using as a probe the bronze gene. And what you can see is the variation in size. It's absolutely amazing. It seems that not a single genotype has the same size in this region. Then they did more studies, and they find that these differences are because the helitron transposons can move genes from one chromosomal location to another. How often does this happen? this is the percentage of contigs that have differences in the number of genes. 0 differences, 25%. 1 to 10 differences, 60%. So there is a huge variation among all the genotypes. I'm just about to finish. So the last thing I want to say is, there is a Brazilian group who did an analysis, a chemical and microarray analysis of lines that have been mutated by chemical mutagenesis or by insertion of tDNAs. I'll be very short. In all cases studied, the observed alterations were more extensive in mutagenized than in transgenic male plants. So chemical mutagenesis cause a lot more differences in the same genotype after mutagenesis than after transformation. So they propose that if we are going to analyze transgenic plants, we should analyze every single input into the food chain, because we don't know what is happening if there are chemical mutations or mutations induced by radiations and so on. So there are a couple of questions there, which I'm not going to read. I just want to end up saying, we are certainly altering evolution. Introducing genes that differ a lot from the maize genome is an alteration of evolution. This is not new. Bloody Mexicans. We domesticated maize 10,000 years ago. The calculations is that in the way we accelerated teosinte evolution, because maize is derived from teosinte, is that within 2,000 years, we accelerated what could have taken teosinte, because it might never have gone into the direction of corn, of 10 million years. 10 million years were reduced to 1,000 years by the intervention of humans in teosinte evolution. Now we are introducing these weird transgenes. Are we ethically and morally prepared to accept the responsibility that we are changing evolution in a different manner of what we have been doing before? That is the ethical question that we have to ask ourselves and assume it if we want to use GM crops. I don't see problems in health. I don't see ecological problems. I don't see problems in the stability of the maize genome. But I see ethical questions that we have to ask ourselves. Are we prepared to assume the responsibility? There is no way back. Once we release this, it's going to be all over and it's irreversible. So I let you with those thoughts to go home and have a couple of sleepless nights. Thank you.
Thank you, Luis. We have a few minutes for questions. Any questions for Luis?
What's your prediction the next 5 years for Mexico in respect of GMA?
Ask me again in 6 months, and I'll tell you. Now it's starting, but the government is waiting for the reaction of mainly Greenpeace. If they get very violent, we have a major problem with these drug cartels now. So the government doesn't want to have extra uncontrollable problems. So I hope nobody from Greenpeace is hearing me here. Yeah. But if they cause a lot of troubles, they might go back and say, no, not transgenic. If not, there are not major protests, it will fully go in Mexico and very rapidly.
Yeah. You mentioned that these new genes or transgenes are integrated by the local farmer. How does this look like? Are these individual farmers who have 2 hectares of land and they select pretty much everybody? Is it the grassroots situation, or are there growers who take the responsibility and distribute seed over several farmers?
No, it's every single farmer individually. But they share their seed. So if they successfully introgress something that is favorable, it will eventually get in the whole region. Because they go to the market and they talk to the other guys and say, how is your crop? And they get seeds and so on. In spite of that, the land races are still alive. So there is a lot of gene flow between different regions, between different towns, and they have been able to maintain the land races for 10,000 years. Although most probably they are not genetically the same as they were 10,000 years ago. Yeah?
What other traits are you working on besides the BT and herbicide, or not you, but Mexican scientists who are transgenic?
Well, in general, in Mexico, we are working mainly on drought tolerance. This is the major problem we have. And the one that's, I was talking to somebody in the morning, that's the killer trait we need. Because if we can show that we can have drought tolerance, that would change public opinion completely. It has nothing to do with the sales of herbicide or insecticides or anything, but it would be a direct benefit to every farmer. 70% of the corn in Mexico is grown under rain-fed conditions, of which 80% has problems of water availability to different degrees, but at least they lose about 30% of the productivity because lack of water. So that would really make a huge difference. I'm not sure whether we can quickly obtain something that makes such a difference, but that's in the direction we are working. Yeah. This is not really official data, but from the transgenic corn that the government purchased in Chihuahua, that it was illegally grown, from the amount of land they grew it, it looks like they had a 15% increase in productivity, although these are not official data. BT and Roundup. They have the most recent lines. So it certainly would have an impact. But the question, the other question is, and this is also a philosophical question. Maize is very related to Mexican culture. And it's been linked to the type of maize they grow. They have certain traditions. They prepare different dishes and so on. What do we do? If we increase productivity in the large farms, we don't need corn anymore. What do we do with these guys? We left them alone to die, or it's a very difficult problem because they don't have very nice standards of living. They have 2 hectares of land, and they produce 2 tons per hectare. Who can live with 4 tons of maize all the year round? Their children cannot go to school because they have to help their parents get rid of weeds by hand. These are true organic farmers. They don't have money to buy herbicides, fertilizers, or anything. But what do we do with them? How can we help them? Or is it ethically acceptable to say, okay, don't grow maize, grow agave to produce tequila? Needs much less water. You can produce up to 80 tons of agave per hectare at 5 pesos kilogram. You make a big difference. But the government hesitates in suggesting that because, okay, the intellectuals are going to complain that we are destroying Mexican culture. A local consumption, unless we make new markets. If we make popular pozole all around the world, like Corona did. I mean, Corona became very popular for a very simple fact. It was the first beer that had a clear bottle. Very successful. Can we make the same for pozole or for tamales? And then increase the price of this type of corn in such a way that now they can make a good earning. That would be the real solution. Not in the internal market, because the people who like to eat pozole and tamales is the lower income people, not the fancy guys in the cities that like to eat sushi and Indian food and Korean specialty food and Russian food in Mexico City or Guadalajara, Monterrey. So it's a difficult problem. I don't know. Drugs and corn are the big problems of Mexico.
And with that, I'm afraid we are running out of time. I would like to thank Luis for developing the tool. If you would like to see him more, he's going to be there tomorrow. If you'd like to talk to him,